All right, so if the previous days were concerned with how neurons talk and how neurons talk to each other, how they connect, how they're able to change their dynamic firing rate with time, which we have seen is the way in which information is encoded in the neuron of the brain. Today, it's going to be mostly concerned with trying to understand of how the brain is going to actually use the information that neurons in different parts of the brain are encoding to do something with it. And do something with it, I mean guide the behavior. We're going to have first a technical part that is about how we actually test the hypothesis. We come out with some hypothesis from our experiment. We want to test it. And this is about manipulating neural activity with light and drugs and other technologies. The second part that is going to be after the break, it's going to be sort of a case study for system neuroscience. That is the long quest for understanding how our brain is able to create, store, and then in last instance recall memories, which is basically the search of the engram. We will borrow an approach that physicists use a lot of times. We're going to look at this question from a historical perspective, how the whole quest for finding the neural correlates of memory have progressed through time. And this is going to bring us in a very speedy way to what I believe is really a new era for neuroscience. It is the one that we are witnessing in the past 10 years or so, in which the combination of multiple technological advancement has made possible approaching questions that were really unthinkable even like 15 years ago. So this is actually a very cool time to be a neuroscientist right now. And it's going to be for the next years. So we are lucky. So again, just a short recap of the thing you should by now know as well as you know your name. That is, the neurons are able to communicate by means of electrical signal, the action potential. One single neuron is not an isolated entity, but integrates information that comes from other neurons in the brain. This information carries a specific weight, which is the way in which neurons are able to select what communication should be strengthened and what communication should be instead weakened through LTP and LTD, so that one single neuron listens to 15,000 other neurons, and only if the message that these neurons are able to transmit is integrated and is able to cross the threshold for action potential at the axonal segment, this neuron is going to propagate information along the network. The last thing that we have seen on Tuesday was that specific connectivity motif allow neuron to change their activity rate over time. This is basically the most important properties of the brain, the fact that the brain is a dynamical system, meaning that this state of the brain changes on a moment-to-moment basis. And on these changes, we can encode things that are relevant for us, like sensory stimuli that come from outside, things that we think internally. We can create substrate for memories that capture the sensory information that we were exposed to during a specific moment in time. The type of change that the neural code allowed are increase of activity, decrease of activity, change in the timing of activity in relation to an underlying metric of the brain, which is the oscillation, and the synchronization of neurons with other neurons in the brain. Of course, this is not the only way in which activity dynamic change with time, but it's definitely some of the most important one. And yesterday, in the end, we saw our Jennifer Aniston neuron. We know that there is two ways to approach the question of how information is encoded, like is represented in the brain. On one hand, we can try to answer the question, how do neurons react when we present a specific sensory stimulus to a subject? And this is the encoding problems. So give the stimulus and try to understand how this changes neural activity. And in the other way, we can go in the other direction, something that we saw yesterday evening. We can look at the activity of the brain and try to understand what stimulus is actually representing that activity. So the decoding problem. Okay? From Monday's lecture and the introduction especially, I told you that in systems neuroscience, we have four main aims. The first one is understanding how information about experience is represented in the electrical and biochemical signal of neurons. This is now what we have called the encoding problem yesterday. Understanding the computational processes by which information is transformed and propagates along neural network. So how you create those neural dynamics that we have been exploring when we were talking about microcircuit motif, and when yesterday we saw the continuous attractor network or the ring attractor, to understand how this information is decoded by the brain and how this is used to support behavior, and then to create a model that can reproduce some of this cognitive function. Today, we're going to understand, try to tackle the question of how in the lab we understand how the brain uses information to link to behavior. In particular, we try to answer basically a question which is at the basis of your system neuroscience research project when you will start to come to the lab and start your PhD with us. That is, you are curious to understand what information is the neural code of neurons in area X represent, and what type of behavior is actually supported by the activity of this neuron. The idea would be that you pick up one area of the brain that you're interested in, that nobody knows exactly what it does. You try to understand what is the information that is encoded in that area, and you try to understand how the other areas of the brain or the whole brain uses this information to actually guide some selective behavior. So in a typical way, what you would do, and you know from the experiment that we have highlighted during this week, is that you would record the activity of the neuron in area X to have any intuition about when this neuron would be active. And you see now that you can use different methods for that. You can use electrophysiology if you are comfortable with recording from a limited number of neurons in acute ways, and to let's say, not be able to follow the activity of this neuron for a long period of time. Or you use calcium imaging if you want to go repeatedly and assess the response of the same neuron over again. For example, as yesterday Maria was showing you, if you want to understand how neurons support learning a spatial map in the water maze task. You quantify the behavior of the mouse during your recording. For example, as Maria again showed you yesterday, you can track the mouse behavioral bouts, the path that is described during his exploration, the latency that it finds to reach a goal, or you can look at specific variables, the amount of time that this animal spends interacting with objects versus interacting with other people and so on. And this becomes crucial because you want to analyze single cell unit, which means you want to align the neural activity of the area that you're recording with the behavior of the animal so that you can quantify correlation between a specific stimulus, for example, as we saw for the play cells yesterday, the fact that the cell was firing every moment that the animal was in a specific location, and the response itself, that is the activity of the neuron. So instead of providing passive stimuli to an animal to understand the tuning curve of the neuron, you want to align and find the variable in the animal's behavior that can explain largely the activity of the neuron itself. At this point, you might use some trick like the decoding algorithm that we have talked yesterday to try to understand if the information in the area that you're studying is, for example, encoded at the level of single neuron and population of neuron, and what are the elements that you really need to pay attention to in order to extract information from the activity of the area, which altogether lead you to one of the most important part of your project, that is the formulation of the hypothesis, which is activity of neuron in area X are important for the function Y. For example, activity of the neuron in the hippocampus are important to create a spatial map that allows you to navigate and find your goal in a task like the Morris water maze. At this point, you need to test that your hypothesis is correct, or your study is going to be a descriptive study, which is extremely valuable and important. And a lot of studies, especially before, I would say, this new golden age of neuroscience, were mostly descriptive in this area because we had crude ways of manipulating activity. Now you can go a step forward and say, ask yourself, and like you really test directly the hypothesis that you have formulated. And ways to manipulate neural activity are many, and they mostly fall into two main categories. You can use manipulation that produce a loss of function by basically asking yourself and wanting to answer in the question or the hypothesis in such a way. You hypothesize that the activity of neuron X is necessary to produce Y, so if you inhibit the activity of X when Y happens, you should be able to basically impair the occurrence of Y. For example, Nuno was mentioning yesterday, if you stimulate the neuron that are able to encode a certain stimulus, a certain sensation, are you able to elicit that sensation? In this case, we should go in the opposite way. So if you're inhibiting those neurons when you're producing the sensory stimulation, Am I not allowed anymore to feel what I would be supposed to feel? This type of the manipulation that induce this type of loss of function are largely lesion. You literally lesion the part of the brain that you think is responsible for that behavior. You can induce silencing of those neurons, and you should have seen one way in which we can give silencing at the molecular level on Monday when we applied TTX to our simulation. You can give genetic ablation of neurotransmission. There is one cool tool in which you block fusion of synaptic vesicles to the presynaptic membrane so that those neurons, even if they receive information, are not able to transmit it to the postsynaptic partners. Or you can use drugs and light to silence neuron. And this is something that we're going to see in the lecture. We're going to go through basically all of this type of technique. The other part of manipulation that you can do is a gain of function manipulation. So if the activity of neuron X is necessary to produce Y, then you should be able, by artificially activating X, to reproduce Y, even in situations in which the stimulus does not occur. We have seen, for example, that the animal, that the neuron is able to respond to a moving bar or an oriented bar in the visual field. With the gain of function manipulation, by activating this neuron, you should elicit the response that is associated to this moving bar, even if you don't present the moving bar in front of the eyes of the animal. To have this type of manipulation, we usually go through a macrostimulation. So we literally stick electrode and activate the activity of neurons. We can have pharmacological activation, and again, optogenetic or chemogenetic stimulation. And this is what we are going to use now. So now we're going to go a little bit through all these technologies. We're going to be talking about each of them. We're going to pay particular attention to optogenetic and chemogenetics, because these are the technological advancement, among the technological advancement that I was telling you before, have made really possible testing hypothesis and performing experiment that until a few years ago was not really possible to address. Lesions are probably the oldest type of manipulation that has been done in the brain and is quite intuitive to understand what it is. These are manipulations that are aimed at eliminating a specific area or population of neuron to assess the impact that this neuron might have on the removed part. By definition, lesions are loss of function manipulation, but of course you can imagine that if you get some sort of specificity for a population of neuron, you can also get a gain of function manipulation. For example, if you are able to target specifically interneurons. Of course, there are many ways to induce lesions in the brain. You have physical lesions. You can have a knife cut, an aspiration. You can inject viruses that kills specific areas of the brain. And these are all methods that have been used in the past. You can have electrolytic lesions that make basically stick an electrode into an area of the brain, induce an excess current, and fry the brain cells to death. This is something that, again, it's something that has been used in the past. You can use chemical, like canic acid, abutanic acid, NBQX, tetanus toxin, or specific chemical compounds that are directed to neurons that express, for example, dopamine, so that when you inject this particular drug topically in an area of the brain, the neuron is in that area of the brain actually die. Or you can have a genetic toolbox to basically express specific receptor for specific toxin, but this is something that we're going to see later how it's working, and it's probably going to be relevant for your study of the sensory, of the motor system next week. We have already encountered a way in which lesion has helped understand how the brain works, and this was the famous case of Henry Molaison. As I was telling you yesterday, this patient had a large resection of the areas of medial temporal lobes that included the hippocampus and some parahippocampal cortex, the entorhinal cortex, to eliminate the foci of epilepsy for this patient, which left him unable to form new memories of personal experiences. He could learn skills, for example, draw a shape. If you are not able to see your hand drawing, he could remember stuff that had happened before the surgery itself. But if you asked him to create a new memory of that of experience after that happened after the lesion, the person animalism was not able anymore. Now, Lesions have been used for a long time, but they have some limitations that are quite obvious. Who wants to start answering one of the most obvious limitations that lesions might have?
It's not reversible.
Yeah, this is a very big limitation. I told you that the brain is plastic. What do you think that the non-reversibility and plasticity of the brain might actually cause, Nuno?
The tissue will try to heal, and healed tissue has a little bit different properties than the one that's surrounding it.
Sorry, I didn't understand because the volume was low.
In the tissue will try to heal the lesion or to compensate for it in some way, and this will interfere with the results.
Exactly. So the tissue will be compensating. The compensation might happen at the area of the lesion yourself. There is axon sprouting, formation of new synapses. That's usually safe things, but not so much. What the most common things that happen is that areas, of course, the brain is smart. You don't have just one area of the brain is the only one that is able to do something. There is a certain efficiency in participating to the same type of phenomenon from other areas of the brain. And you will see this is a phenomenon that is very important, that is called distributed coding. So other areas will take over. So we'll have a compensation with time. But there is a few limitations that are correlated to the manipulation itself that are really upsetting. So I said before, you go with a knife or you go with a toxin or with a drug or whatever you want to do and kill. Do you think you would be able to control what you do in a laser sharp and precise way?
Well, you will cut out or destroy other areas with other functions. So at the end, you cut off your target, but there is a high chance to take off other things that will not be related to the disease.
Exactly, precisely. So in the case of Henry Molaison, for example, the target was eliminating part of the hippocampus, not even the whole thing. What they end up doing, and of course they discovered 60 years after the lesion, because the patient really literally lived for 60 years longer, was that they had eliminated the whole hippocampus, large part of the entorhinal cortex, and large part of the parahippocampal cortex. So the lesion in the end was much larger than what they had anticipated and what they had gone for. And there are multiple reasons for that, of course, because lesions and killing part of the brain tissue induce degeneration also in other part of the brain tissue, because axons travel for very long distance of space, and by cutting them off, you might induce cell death even in the cells that are in another area of the brain, and that sends projection to the area that you're actually lesioning. So yes, the inability, Fabio, to control the size of your lesion is really something that is a great limiting factor of this manipulation. And you basically said all that I was already mentioning here. The second type of, yeah.
Did he lose also his long-term memory, or only lost memory for new things, the short-term memory?
Yes. So the problem is that he lost the ability to form memory in the short term, and those memory could not be consolidated for long term. So you will see one of the doctors that were really dealing with him for a long time was Brenda Milner, who is a remarkable scientist. And please, if you're curious, just read her story. She turned 100 last year, I think. She rocks. He never recognized her. So even if she talked with him and met him on a daily basis almost for decades, she never really went deep into the knowledge. He might form memories associated to something that had emotional valence and so on, but not really like memories. He didn't lose the ability though to remember memories of stuff that had happened before. I mean, he lost a little. It was a little retrograde, the amnesia as well, but it wasn't like retrograde 2 years for infancy, for example, and so on. To the fact, and you will see in the lecture of today, that the area of the brain that they lesion, the hippocampus, is important to convert memory of this experience into something long term, but then it becomes dispensable for those. Okay, the second is a microstimulation. The microstimulation is actually what the word says, so it's very intuitive. You stick an electrode into one part of the brain, and instead of recording something, you actually like inject some current, or you stimulate the cells, so to activate the neurons that are in that specific part of the brain. And because yesterday I've talked to you about the prosthesis, I talked to you one part of the loop, right? When you have your arm, you use it to move stuff, but you also use it for sensations, right? To get some feedback from the fact that you touch or you grasp. That was actually a very difficult thing to control, that people would get like feedback from this prosthesis. Now people are starting to solve this problem again, getting like not only decoding, so using the electrode not only for decoding, but also for encoding something new that is basically like stimulating the part of the sensory cortices that would basically take care of distributing the message. But can you see some immediate limitation of this manipulation? What is the thing that you risk the most when you stick an electrode in a part of tissue blindly?
You can't control which cells you stimulate this way, because you just stimulate the area around the electrode.
Exactly. You have no idea what you're doing. You know that your stimulation is eliciting something, but you absolutely have no idea what the mechanism is. Is this given by the fact that you've activated your exciter neuron? Is this given by the fact that you've activated your inhibitor neuron? The passing fibers, dendrites of neurons that have cell bodies located in an area that is slightly adjacent to the one that you're injecting, it's really blind. So in this configuration, that's the biggest risk. There is another risk, that is you inject too much current, you might induce actually something like epilepsy. So things might be really, really risky. Okay, this is what I was mentioning here. One thing that I just wanted to mention is something that you might have heard and seen already before, is that microstimulation, after a period of, let's say, oblivion, has become extremely relevant lately, not only for the prosthesis that I told you before, but because it is at the forefront of the clinic. So these are microelectrodes that are implanted in the brain of patients that are affected by a different type of pathologies. Now it's successfully using for treating drug-resistant forms of Parkinson's, and is under investigation for other psychopathologies like depression, bipolar disorders, and so on. Basically, the therapy is that you have electrode in the brain and you artificially stimulate neurons in the area where the electrode are by simply like injecting some current. The mechanism by which it works are unknown for the limitation that I've told you before, that we have no idea who we are recording from, but the results are astonishing. And I've just shown you something that basically like affects a person affected by Parkinson. So the results are really black and white. We don't know really why. I mean, the aim is to stimulate the part of the brain that is understimulated because of the loss of the dopamine neurons. But the results are really like changing the life of the people that are subjected to this type of treatment. So it's actually very nice for the clinic. Okay, we go to pharmacology. Pharmacology is a very powerful mean to manipulate cell activity. As you might imagine, that basically like consists in infusion of specific drugs that are aiming at activating or inactivating specific ion channels, neurotransmitter receptor, ion pump, etc. This is the way in which the manipulation that you've done Monday in your simulator could actually become true in vivo. You might work on blocking, for example, some sort of currents, the voltage-dependent gated channels and so on, to boost or decrease the activity of specific area, specific population. What may be the advantages and disadvantages by the pharmacological manipulation, and what is the biggest risk? One, yeah, but this actually is a very, like, because we know a lot about the channels that are expressed by different subpopulation of neurons, this was a disadvantage before, and you have some drugs that work on everyone, but now it's actually less, because we really can go and target specifically some channels apart from others. For example, when you use diazepam, you're going to target specifically one specific channel for GABA that is expressed by some specific population of neurons. So this can actually be overcome with modern techniques. But there is one more immediate advantage in this case, that is something that is referred to what Nuno was saying before, Someone remembers what just happened?
It is not invasive. One does not have to implant anything in the brain. You just administrate it via maybe an IV injection or into the cerebrospinal fluid if it can't pass the barrier.
This is definitely an advantage, but you need, in that case, you need a very specific drug, right? You have two problems that I can tell you are real for the pharmacology. Is that one, you need to cross the blood-brain barrier, which is something that is not trivial. A lot of molecules cannot cross that. And the second is, if you have a drug that is extremely specific, that's perfect. If you have a drug that is not as specific, in that case, you might target all the different nuclei of the brain and create something that is very dirty.
Isn't there also an issue of like the concentration of the drug in the body and how it is metabolized? Because depending on that, the amount will start to.
Absolutely. This is something that is correlated to what the biggest risk is that you might overdose and you might actually like induce something that really is more like epilepsy or catatonia. And from catatonia, you can recover when the drug is cleared out. From epilepsy, many times you can't, the brain is fried. Absolutely. But again, in this case, you need to do some thorough study. That is what pharmacological company like Novartis and Roche do to understand, the clearance of the drug from the brain, the concentration at which it works, and so on and so forth. But let's focus on the advantage. What is one of the advantages that a drug has?
It is reversible instead of the lesion, for example.
Exactly. So you can inject the drug, and you can see the effect, and then that helps you formulate the hypothesis. The drug then washes out. At a certain point, the effect is gone, and you can actually test if the function comes back. So in an end, you don't have something with the lesion. It's more acute, so the effect is only when you give the drug, or most of the time is only when you give the drug, and you can actually overcome problems like the one of compensation that I told you before. So yeah, it is a reversible manipulation. Do not bind it irreversibly to the target. It can be specific to subpopulation. Again, this is something that we just discussed. And usually the most frequently used manipulation is to induce a reversible inhibition induced by mucimol or lidocaine. So basically these are two drugs that inhibit excitatory activity, and you're able to silence a population of neuron, which is by far the safest thing because you cannot induce really like epilepsy if you target the drug right. And you can see if when you give the drug, the function, the neural code changes, the function changes, and so on. For example, one thing interesting that I can tell you one very cool study that was done on grid cells was that they recorded grid cells while the hippocampus was inactivated by the infusion of mucimol. And what they saw is that grid cells would start to change their tuning and become head direction cells. And then when the mucimol would wash off, they would go back to fire with the grid pattern, which it's again predicted from the continuous attractor model that I told you yesterday. But if you're interested in knowing more about this, just let me know and I'll send you the reference. Now we go into a class of different type of manipulation, which are quite specific to subpopulation and that use gene expression and as a way to target your manipulation to a specific class of neuron that express specific gene. One very ingenious is the use of toxins. This is something that you will see next week, and probably you already have read a paper that basically uses this. This is very ingenious. If you have a specific promoter for a cell population, you can induce that population of neuron to express the receptor for a toxin that normally would not really bind to cell unless there was a receptor expressed by the neurons. And then you can inject the toxin so that only the neuron that expressed the receptor will be able to bind this toxin, internalize it, and induce cell death. So this is actually very a nice way to kill a specific population of neuron and test the impact that that population of neuron has on behavior. Again, after you've done your manipulation. So this is the schema here. You need to basically have a cDNA or a construct that induce the cells of interest to express the receptor for the toxin. If you inject the toxin into a wild-type animal, you have no effect. If you inject the toxin in an animal in which a population of neurons express the receptor for the toxin, then you kill that population of neurons. But there have been two different types of manipulation, always based on exogenous gene expression, which have become really fundamental for trying to understand how some population of neurons support behavior. The first of this population is optogenetics. Optogenetics is a technique by which you use light to stimulate neurons. And you can use that. How many of you have already heard about this? Okay, so I'll go and express. So we have talked about photoreceptor in your eyes, right? We have talked about the rods and the cones that are able to pick up light and transform light into an electrical signal. And when we saw this on Tuesday, I think, we also discussed and said that there were proteins, we said that these neurons were able to sense light around, and the ability for them to sense light is because they have proteins in their membrane that are able to be excited by light and basically like induce a conformational change into this protein or the activation of the protein that in last instance results in opening and channel depolarization or hyperpolarization of the membrane. So these neurons these proteins really allow neurons or cells in general to sense the environment around them, in particular sense the presence of light in the environment around them, and response to light with, in last instance, the opening in a channel that change the potential of the membrane. Now, there is particularly a class of opsin. These are expressed in microbes, which are used for photosynthesis, that are very particular because they're quickly open by specific lights at specific wavelengths, which are not the one that we see, because bacteria use a lot of different wavelengths. They live in places that don't have the visible light that we see, but they're also able to immediately reisomerize back to the closed configuration when the light is withdrawn. So they're very fast in switching on and off based on the presence of the absence of light. And these proteins have become the substrate of heating efforts that have made them able to respond to desired wavelength, that are wavelength that normally we would not see in the light around us, and are able to become permeable of selective ions, and specifically to sodium, potassium ions, and chloride ions, so that we can basically be able to tailor this protein to induce the activation or inhibition of a neuron by just giving the light to the appropriate wavelength. We have basically two main classes of this protein, the one that get excited by light and respond by an increase in permeability to sodium and potassium, so that basically you have an increase of excitation of the neurons, or neurons that respond by increasing permeability of the membrane to chloride, so you have the inhibition of the neurons. We talk about channelrhodopsin to define basically like a protein whose activation by a blue light, which is around 450 nanometers in wavelength, is able to make the membrane permeable and increase and depolarize the cell. An inhibitor channelrhodopsin that instead has the opposite effect, same wavelength of light, but hyperpolarization as the effect. Halorhodopsin, which is instead a protein that is able to be turned on by light at different wavelength and induce again inhibition of neurons. Or archirhodopsin, which works with protons instead of working just with other type of ions like the one that we have seen before, that has the same effect. So activated by light and able to basically induce the hyperpolarization of the membrane. These proteins have been engineered in a way that now are quite reliable in being able to induce activity of neurons with a very short temporal delay. This is a neurons that I recorded during my postdoc. You give a stimulus of light time zero, you see that with a latency of more or less 2.7 milliseconds for different trials, you get a response from the neuron, which is a spike from the neuron that expresses channelrhodopsin. Okay? So if you express one of these proteins in your neurons and you deliver the light at the appropriate wavelength, you are able to activate and induce a response that is basically following your stimulation. Of course, these, as I said on Tuesday, the brain is quite a fat tissue, so light cannot really penetrate very deep into the brain if you give it from the surface of the brain, especially not at this wavelength. There is the skull, so it doesn't really pass through the skull. So the complication is that you need to create basically let's say an implant into the brain of the animal with an optic fiber that is able to deliver the light very close to the area that you want to stimulate. In this case, let's suppose that we want to stimulate neuron in the CA3A of the hippocampus that is here. I need to have this neuron express the channelrhodopsin channel, and then an optic fiber that is implanted to the very, very close to CA3, as people have done in this rat. The effects, though, are astonishing. Imagine that you are able to identify a population of neurons that you are really hypothesizing are involved in the control of aggression. You will see now that by optogenetically stimulating this neuron, you can actually look and see if you're able to reproduce the type of behavior that you're actually studying, or that you have hypothesized depends on the activity of this neuron. So these are some quite general public movies, but they're basically like papers right now are quite full of this type of studies. In these studies, you have an area of the brain in which people hypothesize there were neurons that, when active, were basically responsible for activating aggressive response in mice. You have, wait a second here, that the neurons that were hypothesized to be responsible for aggression in mice were actually transduced with channelrhodopsin, so they express channelrhodopsin. The optic fiber was implanted very close to this neuron, so that you can formulate the hypothesis that if those neurons are really necessary to induce aggression, the synthetic activation or the artificial activation of those neurons should induce the animal to become aggressive. So to become aggressive, they put a cricket to basically have something to target the aggression. They put an object in the cage of the animal. And you see here that in the previous type of manipulation, let me go back here, when the laser is off, the animal is running around in its cage, don't pay particular attention to the intruder that is the cricket. But as soon as the laser comes on, then the animal is immediately going and attacking the cricket itself. And you see something now that even when an inanimate object is in the cage, the animal doesn't care. As soon as the light goes on, the attacking response is actually elicited, right? Okay, so this was the new scientist type of general public association. Another type of behavior was you find area in the hypothalamus which you suspect are important for inducing eating responses in this animal. You want to test if the neurons, when you artificially activate those neurons, if those neurons are really responsive for the feeding behavior, you should be inducing the animal to go and eat. This is what you do. The animal is running, is close to food, you activate the light, the animal starts eating. And these are very well-fed animals that normally would not really spend too much time investigating or interacting with food. The light goes off, the animal starts to lose interest and doesn't eat anymore. The light goes on again, the animal goes, finds a piece of food, and he starts eating. So this is really a very, very fundamental and revolutionizing tool by which we can really investigate, or if you want to test, the involvement of specific neuron at a cellular basis onto specific behavior. And in the papers of today, we're going to see basically like an example on these on trying to recall and reactivate memories. Here again, I want to just point out to the fact that yesterday our discussion on LTP, it's not the stimulation on its own, the light, but really it's the identity of the neuron that express the channelrhodopsin, so the activation of the specific neuron that matters. If you activate a population of neurons which you are maybe in an area that is close by and you not suspect is involved in the same type of behavior, you don't find the same type of effect. So always protect your control wisely. Of course, though, channelrhodopsin is very precise, as you see, it's very powerful, but it's also quite invasive. You need to implant an optic fiber in the brain of the animal, and you also, as you see here, you have a continuous cable that tethers the optic fiber to a laser source, so that you can give the stimulation at the proper wavelength and power. Around 10 years ago, maybe like 12 years ago, a new tool has been invented. It is chemogenetics, in which you chemically engineer some channels that would be, or some receptors that would be expressed by neuron in general to be sensitive to one specific ligand, so that usually would be inert in the body of the animal. So that when you have neurons express that modified receptor or channel, and you give the ligand, you can induce a chemical binding of the ligand to the channel or to the receptor and change the excitability of the membrane toward depolarization or hyperpolarization. I will show you what happens here. This is the first type of molecules that was developed for this use. It's called DREAD. The design receptor expressively activated by designer drugs. That's the acronym means. These are channels that are in large part actually second, like a G protein coupled receptor. And then they bind a molecule that is called CNO, which would otherwise be inert in the brain of the animal, although we see one caveat in a moment. And only in cells that express the channel, and only when CNO is injected or present in the brain of the animal, CNO will bind the specific receptor and will induce either the increase in calcium concentration within the cell, which is basically inducing activation of this neuron, or decreasing the calcium concentration in the cell, or AMP, cyclic AMP, so that we basically have an increase in excitability or a decrease in excitability, which produce a heightened response or a lower response from your neuron. The problem comes that it has been discovered quite recently that CNO on its own gets metabolized by our liver into a molecule that is actually active on the endogenous channels. So this is a big confound of the technology, which means that you always need to pair to the experiment in which you have one class heteroimol, in which you have the channel and you inject the ligand. You always have to have a control in which you inject the drug and you see that the injection on the drug on its own is not sufficient to give your behavioral response. I was mentioning to you previously during this week that finding the best, like the right control is one of the most important things that you do during your project. So, for example, we have here two channels, HM3D, we increase calcium and increase excitability, so the neuron fires more action potential when you give CNO, or HM4DI, which decrease AMP, cyclic AMP, and decrease excitability of the neuron, so the neuron fires less action potential. There is another class of these chemogenetics, which is directly coupled with basically channels instead of G protein coupled receptor, which works basically in the same way. You give a drug, PSEM in this case, that drug is able to bind to the modified channel that is called PSAM, and is able to induce either the excitation of the neuron or the inhibition of the neuron. In this case, though, instead of being an indirect way in which stimulate the neuron, this is a very direct way in which you can actually do stimulation or inhibition. So now we have these two big class of methodologies, which are fundamental, and then both of them you probably will see or have seen in the paper that you're studying today. You will definitely see in the lecture about the engram. When you use optogenetics and when you use chemogenetics to activate or inhibit your neuron. With optogenetic, of course, there is a very big advantage that is very good for a short period of time, this is actually the disadvantage. The advantage comes after. You're shooting a laser into the brain of the animal, and you're shooting a laser into the brain of the animal at a wavelength that carries a lot of energy on its own. It's not like the two photon, that unless you have the two photon at the same time, the neurons are, the molecules are actually, electrons are actually not excited. At this wavelength, electrons that are excitable are actually excited, which means that you will overheat the brain. You will overstimulate molecules that are in the brain, like water and other stuff, and you basically overheat it. So you should always use optogenetic for short periods of time, but it's bad if you want to stimulate an area of the brain for like half an hour. This is a problem that Maria knows very well. She has been trying to use a different wavelength to do an experiment in which you label some neurons, and basically like the first trial of this were actually creating too much of a damage. Chemogenetic instead is good for entire behavior session. You inject a drug, most of the drugs actually work for half an hour to a couple of hours, so you have a very long and inhibition of or excitation of this neuron. The problem is, of course, that optogenetic is you can activate and deactivate in terms of milliseconds, which means that it gives you a strong temporal precision in the dynamic in which you activate or inhibit your neurons, while chemogenetics has a very low temporal resolution. Optogenetics needs more equipment, and the animals require the tether. You have a cable, which might impede some sort of behavior, while chemogenetic requires less equipment. The animals move freely. You can test more animals simultaneously because you don't need to connect to the laser. So you can really easily apply it for large-scale experiments. optogenetic is best for small brain regions. And I want to ask you, because you should know, why is it good for small brain region?
You can, with the light, you can manipulate small regions by changing the intensity.
Mm-hmm. Why can you can manipulate only small brain region?
Well, this is now a guess, but you can change the intensity of the light, and therefore it will go deeper into the tissue or not. And also the light itself is limited, while with the chemicals you target mostly the whole brain. And then it will be difficult to target specific regions.
So this is part, you answered the question when you said that you cannot reach far away region, right? Light really doesn't travel very well in the fat tissue. So at a certain point, you basically have that light will reach just a few hundred micrometers around the optic fiber that you implant. You can increase the power, as you said before. The problem is that if you increase the power of the laser, you also increase the amount of energy that you're giving to the brain, and you're also increasing the damage that you create just by giving that particular power. So it's always a trade-off. And this is also the same type of trade-off that you have with imaging experiment, in which you need to tailor the power of the laser that you give to the brain to be enough to do what you want to do, but not too much in order not to destroy the tissue. So yes, the answer was indeed that, light doesn't travel that much. Well, instead, if you have the expression of your receptor in many different areas of the brain, and you give a drug, all the different areas of the brain can be targeted at the same time if you just inject the ligand, because it's distributed. Okay, so you can do a precise manipulation. For example, if you have a complex task, you can, for example, like inhibit the activity of the neuron just at a specific task. Like imagine that you want the animal has to learn to wait for a stimulus and then press the lever. You can inhibit the neuron specifically when the stimulus is delivered or when the animal needs to prepare to press the lever. So you can test the function of a specific population at a specific time point of a complex behavioral routine, while chemogenetics is less precise. But the advantage of having chemogenetics, especially in the DREAD form, is what I was telling you before. Because DREAD works on excitability of the neuron, with optogenetics, it's like throwing a grenade into the brain. You shine the laser, you activate or inhibit all the neurons that express the channel. You have no way of creating this in a more physiological way. What you're doing with chemogenetics, you can tinker the way you give the drug or the amount of drug that you give in a way that you basically just make the animal more or less excitable, but still they're excited by endogenous excitation. So it's much more close to the natural way in which neurons get excited than to the completely artificial way to which neurons get excited with optogenetics. And this is very important from what I told you yesterday. The way in which neurons are activated in time is the fundamental way in which basically the brain encodes information. So optogenetic is much more artificial, if you want, as a manipulation than what the brain does normally. Yes, so you can have, because you can basically like have, again, this temporal precision, optogenetic is very good if you want to manipulate activity within the same animal, within the same task at multiple time, or you can even like think about activating at one phase of the task and inhibiting on the other phase of the task, while when you inject the drug with chemogenetics, you basically elicit the response and you have no control on the temporal delay. Yeah, well, this is basically like the data from the optogenetic are easy to interpret because, again, the stimulation can be so short that you are quite sure that no compensatory effect happens during the time window in which you've actually given the stimulation, while with optogenetic, this is much more difficult. Okay, one thing that has become clear from the last, from this last slide, and this is really literally the last few slides that you have, is that getting to manipulate neuron by providing exogenous payload proteins that are, for example, channelopsin or the DREAD receptor or the toxin receptor, is allow us to combine the ability to target specific subpopulation of neurons that express specific genes with manipulating or recording their activity, which has created what I was telling you before. What we are witnessing right now is an explosion of knowledge about the brain that is comparable only to my favorite decades of the past century, that is the 50s. In the 50s, there were all the ideas of Hebs coming along, the experiment from Hubel and Wiesel coming along, all, HM was also done in the 50s. That really propelled our knowledge of the brain immensely. And we are witnessing the same thing right now. Because at this moment, by using a specific gene and gene expressed by specific subpopulation of neurons, and the way in which we can record activity from individual population of neurons with calcium imaging and manipulating them with chemogenetic and optogenetics, we're really able to gain a unprecedented detail in the way in which the brain works. Defining a subpopulation is not easy. You have had this discussion last week during the course, right? You can divide a population based on many different criteria. That is when the neurons were born, from which mothers they were born, if you're talking about clones, at what time they were born, who they connect to, both locally or long range, what is their intrinsic gene expression that is an influence on the intrinsic properties like excitability and so on, or their function, right? The firing properties that we've seen yesterday. We have divided a population of head direction cells, place cells, grid cells, and so on. If you can find the promoter, an enhancer, an intersectional strategy, or a viral tropism, that is the susceptibility to be infected by a specific virus that targets one specific subpopulation, you can record from that specific subpopulation, and you can manipulate the activity of the specific subpopulation to study the contribution to behavior. And this is really what propelled in the last 10 years, 15 years, our knowledge of the brain enormously. And this is why institutes like the Allen Brain Institute in Seattle are really investing resources that are billions and billions of euros, dollar, whatever you want to call it, to actually try to map the cells of the brain, to map cells at the level of what gene they express, what projection they have, and what type of firing activity they have during naturalistic behavior. Most of this is based on the most of our ability to target specific population is based on the recombinase system, like the Cre or LOXP, that allow us to express specific genes in specific population of neurons. There are two ways in which you can express an exogenous protein with Cre and LOXP. I don't go into detail because you have heard about this extensively during the course of study. You can either get the inversion of the transgene so that it goes into the right orientation for translation, or you can have a stop codon before the operating frame of the gene so that the gene cannot be expressed. One thing that I just want to highlight, because it's going to be extremely important for the future, is that for the future lecture, not for your future, maybe also for your future, is that there is a version, an engineered version of Cre, which has been fused with the ERT2 part of the estrogen receptor, which allow us not only to control who expresses that particular protein by a gene, but also when that particular Cre is going to be active. Have you ever heard about the Cre-RT2 system and tamoxifen? Okay, so I don't see many heads, so I really need you to learn to understand this. So the protein has been basically fused with a part of the estrogen receptor, which is the part of the receptor that is necessary to keep the receptor in the cytosol and in close proximity to the membrane. What it means is that a protein Cre fused to this particular part element of the estrogen receptor protein is not in the nucleus, where it can interact with DNA and where it's necessary to actually induce the recombination, but is sequestered in the cytosol. So it's physically separated by the substrate of the recombination, which is your DNA. So it's not enough in this configuration to actually have the protein expressed, because the protein will not going to be in the cellular compartment where it has to do this action. You also need to provide tamoxifen to the cell in order for the Cre to be active and to induce recombination, because tamoxifen is going to bind to this ER element and induce the translocation of the Cre-ER protein into the nucleus, where it can induce recombination. So you see, you have an intersectional strategy which is extremely powerful, and you see it later in the case of the engram, by which you can select which population of neurons express your gene, your transgene by selecting a cell-specific promoter that target that specific subpopulation of neurons. Only that specific subpopulation of cells we have the CRE-ER protein in their cytosol. But you can also select when that recombination must happen by injecting tamoxifen in a specific moment. And you will see how this will be really the way in which you can target population of neurons that are active at a specific time during experience. In the next lecture, though. The last thing that I want to add, so first of all, is this clear? Because this is really going to be important for you to understand how we found the engram. So we, neuroscience, I mean. All right. The last thing that I want to say is even, and send you in the lab with this thing printed in your brain, even manipulation are not risk-free. You really need to, have the experiment or your conclusions replicated with other means of technology, because every one of these technology, as you have seen, has positive point and negative point, and the risk of overinterpreting your data is real, because you might have that it's actually not the population that you have manipulated that is responsible for the behavioral response, but is another population of neuron which is controlled by the population of neurons that you have actually manipulated, and so on and so forth. And this is a cautionary joke about how science is really prone to overinterpretation, and how replication by you, but also by other lab, is the ultimate validation. The only way to have reproducible science, the only way to have real conclusions and truth is to be able to have it replicated by other laboratories. So we should all strive and work for allowing other people in the world to replicate or to challenge our results. So please always strive for reproducibility, document your experiment well, especially in papers. Don't be smoky about what you do just because you don't want other people to do it, and be open about the data. In last instance, having published a paper is good, is the first step. It's not the end of the story for that specific question. That conclusion must be replicated for others to stick, otherwise it's going to be lost in the wrinkle of time. And that's it for this part of the lecture. We're going to have a break, and we're going to talk about engram in half an hour from now. So, is there any question about this part? Any comment, curiosity, application that you have in mind, and that you just want to share or suggest for some of our experiment that we do in the lab? Okay, so I will check now Slido if there is any question. Oh, there's a lot of questions. Okay, are specific? I'll start in order, right? Are specific types of neuron, like unipolar, multipolar neuron, concentrated a specific area of the brain because of the functions of these areas? Yes. So this is exactly. I mean, yes. The response is exactly that. Unipolar neurons and multipolar neurons are neurons that are basically different conformations, or different, if you want, arrangement of dendrite and axon and so on. I think we can safely say that the neuron that you find in the area are really evolutionarily optimized to the function that that area does, right? So, for example, one clear example is the example that I show you from the sensory neurons, right? The sensory neurons have a dendritic arbor that is connected to an axon that goes to the cell body, that is connected to another axon that goes away. And both the two axonal sides are actually myelinated, which is the way in which information flows faster along axon. That is specific, basically like anatomical configuration that is very efficient for transmitting very fast information that comes from the senses. And that's why you find it in sensory epithelia. So yeah. Okay, so the second one is also with lesion. I think you don't really get information about the role of molecular components that drive that function. Of course, this is absolutely right. In that case, you would need to if you're thinking about molecular components as, for example, receptors that are involved in a function, or intracellular pathway that are involved in explicating the function. You're not asking about that at all. You're only asking, in the case of the most crude manipulation or lesion, if neurons that are located in a specific area of the brain are actually important from that specific function. At the molecular level, you get basically no sense. But it's the first step, right? Because you know that those neurons are involved in that behavioral function, and then you can go in, zoom in on those neurons or that area, and do genetic manipulation selectively in that area that are basically like to understand the role of that specific molecule into the behavior. If this was the question, right, that you want to link molecules to behavior instead of neurons to behavior. One example is in the hippocampus. It was known that the hippocampus was used to episodic memory. We'll see it in a moment. It was known that an NMDA receptor for glutamate were important for inducing this LTP phenomenon that we have discussed. A group in Boston, Tonegawa, went and knocked down the main subunit of the NMDA receptor in a specific area of the hippocampus, and found out that the expression of NMDA in those neurons was necessary for one specific function, that was to separate memory to separate similar memories that are still two individual instances of the same event, for example. So that was clearly one way in which the first studies of the lesion guided a molecular approach to understand the function of a protein to behavior. So with drugs to the limited localization, it could be hard to tell if the effect you see is indeed caused by manipulating the desired cells in a brain area. Absolutely, you need to have a so you need to put two things together. In that case, something that you can do in animal studies, of course, you need to deliver the drug. If, let's say, let me say this first. If you're targeting a receptor or a molecule with your drug that is expressed by multiple population of neurons in multiple areas of the brain, you cannot just inject systemically and say that the effect is given by that specific area. You can say that the effect is given by some of the neurons that express that receptor, but not that specific area. One thing that you can do is that you can implant a small cannula that ends in the area that you want, and you can go and inject the drug directly in that specific brain, which is, again, it's not extremely precise because you know how laterally the drug diffuses, but you can still confine the localization of the drug that you're giving to one specific area. So yes, to your concern, but there are ways to overcome that. Can drug abuse be a disadvantage of pharmacological treatment? Yes, if you do it in humans. And I think what we are assisting now with the US is a clear example of what not to do, in the sense, or let's say, I've been harsh with that comment, but I don't know if you know about this opioid crisis in the US. So there were some drugs derived by opioids that were used for the treatment of chronic depression. These, of course, are recreative substances as well. People become addicted to those substances, and in the moment in which they stop using the substance to treat their chronic pain or their pain that comes from whatever they do, but they start using substances just to recreate and become addicted to that. And a lot of people every year die in the US exactly for that. They were taking a drug for curing something, and then they become addicted to the drug. It escalates sometimes and goes to heroin. So yes, this is definitely a concern. What do you do with the mice when you do not need them anymore after the experiment? We humanely sacrifice them, which means that you basically deeply anesthetize them to levels that you don't get anymore any reflex from the animal or pain responses from the animal, and you perfuse them, which means you inject, or let's say inject paraformaldehyde, which is a fixative solution, into the body of the animal so that the brain can be actually fixed. So you can basically extract, you can slice it, you can make stains, you can, for example, every time you implant something in the brain, you need to verify that your implant is in the correct position. Every time you express some exogenous protein, you have to verify that the protein was expressed in the neuron that you think it's expressed. You have to verify that you have not created too much of a lesion with your experiment. And you can study with immunohistochemistry, which I think is something that you have already seen before during other blog courses. If your manipulation has induced an expression of a certain protein, change in the conformation of certain certain molecules. If you have different antibodies that are specifically recognizing, for example, the phosphorylated form of the protein versus the non-phosphorylated form. So in the end, the experiment doesn't end when you don't need the animal anymore. You perfuse it, you extract the brain, you continue to investigate most of the time at the molecular level how the brain has reacted to your manipulation. How does light induce the conformational change in the opsin? Well, it's about excitation. Again, you basically transfer energy, which means that you can break or create bonds between the atoms that are basically in your protein, so that you create different type of connection at that moment that give you the three-dimensional structure of the protein, and the three-dimensional structure of the protein changes. Once you change the three-dimensional structure of the protein, we have seen with GCAMP on Tuesday, you can bring, for example, two parts of the GFP, which normally would be close to each other, but we have engineered to be far away from each other. You can bring them close to each other again, so that when the conformation change happens, the protein is able to absorb and emit fluorescence, so photons. So this is basically just a conformation change given by the increase in energy that you give to the protein itself. Do you see the optogenetic technology being applied to therapeutics in the future? I don't see it applied in the future. I see it applied in the past. This is already happening. I mean, Boton Droska in the IOB is leading a task force for trying to use optogenetics to restore vision. Mainly, I told you that like opsins are used to excite rod and cones for vision. One of the intuition was that you can actually use it to, you can actually express some artificial opsin if the photoreceptor degenerate in the second station of the retina, induce the activation of these cells with a laser that be able to translate the image that you see into a pattern of laser activation, and you can stimulate the retina with the laser instead of stimulating with the normal light that would come from the outside. This is something that is going on in many different ways. We have the local hero, which is Boton. We have also other people in the US that are trying to do the same. It's a little bit more difficult to do deep within the brain, as you see, but I've talked to you as optogenetics. Optogenetics was basically the first one that maybe was really using an exogenous way to manipulate activity of neurons based on the expression of transgene. After that, there is a lot of other technologies that are being developed. One that was extremely promising was magnetogenetics, that is using magnetic fields to induce the opening of channels. Exogenous proteins, again, in the same principle, opening the channel and induce activity. You have two problems there. One, you have to have the neurons express the transgene. Two, you have to have a way to deliver the stimulus that opens the channel. If we solve these two problems, they can be applied easily. Can you use 2-photon in combination with optogenetic to, for example, excite a subset of neuron like an engram in the brain? Yes. And this is something that it's not trivial, because of a problem that you might not have anticipated, that even if you're extremely precise with your 2-photon lasers, the probability to excite neurons that are close by is very high. So you have to come up with a way to move the laser and be extremely precise at the cellular level in the stimulation that they're doing with optogenetics. So there have been a lot of development in the past 13 years. The first time I heard about it was 2007, yeah, in which people have developed way to move the laser or target the laser precisely with a single cell precision. Right now, before it was sci-fi, right now it's actually reality. The microscope that I showed you before, the one that we have in the lab, is ready for that. We have a second laser path for inducing optogenetic, 2-photon optogenetic stimulation in combination with imaging, and we are going to be setting up soon, hopefully. This is planned for when we move to the new building. So in that case, we are going to be able to record from a population of neurons and decide to stimulate some that have an interesting pattern of activity, or some, as you mentioned before, that are in the engram. Are chemogenetic activator overexpressed compared to the natural GPCRs? That depends on you. meaning that it depends on the virus that you use and on the promoter that you use for their expression. They can be expressed at physiological level if you use a promoter that is expressed at physiological level. They can be expressed in excess if you use promoters that are constitutive like CMV or other type of promoter like cytomegalovirus promoter that boost a lot. There are other viruses whose expression is picking up with time, and if you use those viruses to express your DREAD receptor, yeah, you get to be overexpressed. Did they purposely remove more than just the hippocampus, or did they have to do so due to the complication? This is a good question to which I don't think that I know the answer to. The point is that in the 50s, they really didn't have much of a technology to know where they were going, right? To see the brain in real time. This is actually a big problem today as well. When you use your machine to visualize the brain, you have a millimeter precision in most of the time, unless you use like the most sophisticated methodologies that have a magnetic, because you use the magnetic field to visualize the mostly water atoms in your brain to actually see transition, phase transition, right? And you get the black and white image that you know from the TV, or if you did a scan yourself. At that moment, they didn't have that. Plus, the anatomy was not so precisely defined in humans to say, you know, this is just the area that you need to remove. You have to do exactly the cut here. I just want to show you something quite interesting. That is that in the past, it has been, people have tried to use some expression of fluorescent proteins in use with viruses to try to visualize exactly where you have to do the cut in the brain or in a tissue. For example, if you have a tumor and you have to, it's the same problem of tumor resection, right? You don't know where you actually have to resect. You don't know where the cells stop. You don't have that type of resolution. People have started to try to express a fluorescent protein to visualize the part that they want to cut, which should supposedly to express the fluorescent proteins to be quite clean, especially with tumor problems. But if they remove more on purpose or not, there might have been complication. There might have been, you know, physical condition. You don't want to cut vessels in the brain. I honestly don't know. Can you have subcellular precision with the lasers and optogenetics? Yes, you can stimulate just individual spines. Definitely individual stretches of spines. I think also individual spines. That actually was a quite cool study that I can recall right now, in which they have shown that you activate spines that are close by in a sequence. The summation of the depolarization that these individual spines would give is much higher than what you would actually predict. So dendrites are amazing things, not only because of the complexity they can reach and that we have discussed before, but also because they are very active ways to amplify messages if those messages are arranged in specific ways. So a lot of questions about this. Nice. Any other curiosity or question that might have arisen, or something that wasn't clear from my answer to these questions? Okay, so we have only 15 minutes now, but we'll see each other at 11.